Hi, everyone. My name is Wang. I'm a PhD student from Dr. Mike Beer's lab. And today it's a great pleasure for me to share on this symposium about my research towards understanding the 3D genome organization. And my title is Loop Extrusion Model Predicts CTCF Interaction Specificity. So it's been abundantly clear over the past decade that the 3D genome organization is not randomly distributed. It's actually intimately associated with transcriptional regulation. The 3D organization of genome has been extensively profiled by Hi-C and CHIAPAD, and they identified structural units like topological domains and loops from the whole genome. So the most prominent feature of TED domains is that they are usually bounded by CTCF binding sites at both sides. However, it's not clear what determines the interaction specificity between those CTCFs. And to study that, we built a mathematical model from the loop extrusion process. So briefly, the loop extrusion process says for a loop to be formed, the CTCF sites has to be bound by CTCF at both ends. Then cohesion is being loaded in between them and starts the process of translocation. And when it meets CTCF from both ends, cohesion is blocked and the loop is formed. And what we did here is to transform this biological process into a mathematical model by explicitly modeling the probability of CTCF binding, the decay of contact frequency dependent on distance, the loop competition of existing and new CTCF pairs. And finally, we'll put all those together into one equation by taking their product. We got the probability of CTCF interaction. By comparing our prediction probability with CHIAPAD CTCF, the CTCF CHIAPAD data, we found that our prediction is highly similar to experimental results evaluated on either AUPRC or correlation. And here shows a concrete example of their similarity. We displayed the experimental interactions and the predicted interactions on the heat map and on the genome browser track. As you can see, they're very similar to each other. And finally, we want to ask whether loop competition or distance is the most important factor to determine loop formation. And to study that, what we did is to control one of the two factors at a time. And what we find is when distance is being controlled, loop competition is still predictive of the specificity of CTCF loop interaction. However, when loop competition is being controlled, distance itself is not predictive at all. Therefore, loop competition is more important for loop formation than distance. So to summarize, what we find is that CTCF is one of the most important players in shaping 3D genome architecture. And the loop extrusion model is capable of accurately predicting CTCF interactions. And finally, loop competition instead of distance is responsible for determining interaction specificity. Thank you.